All right, take two. Hello, everyone. I hope you can hear me. I reset the session because something was messed up there. All right, please thumbs up that you can hear me, see me, etc. Good. Let's wait a bit. I will get Frank on stage. So people are joining. It didn't opt me this time to pull everyone, so I'll just see if there is an option to pull everyone. Yeah, we'll start in a minute. I'm just letting people come in case there was a confusion. Alright, I'm gonna try to invite Frank. Hopefully it wouldn't mess anything this time.
Hello.
Hey, Frank.
You can hear me all right?
Yes. Can you hear me?
Awesome.
Good. Alright, so hello everyone. We'll start and essentially today you're gonna hear a lot about neuronal circuits and networks and what they do. So you heard a lot of interesting things in the past days, and you'll see how quite a few of them connect to that topic. Let me share the screen. All right, presentation. All right, so for now I'm not seeing the messages. Frank will let me know if there is anything I should be aware of. Frank, can you say if it's full and visible?
Yeah, everything looks good.
All right. So welcome everyone to day 4. We'll be talking about simulating computational models of brain microcircuits and their signals in mental health. Just a few reminders. Please feel free to ask questions during the session. You can put them in the chat box. I will pause at a few places during the presentation to take questions and answer them. GatherTown is a very cool virtual space that you can meet us and also ask questions and interact, especially after the talk. If you haven't done that, then restreaming the videos is really useful in case you miss something or you want to see something again that you didn't understand the first time. And fill the review about how the session went. And here is the email in case you need to contact us with any other questions. I'll also again acknowledge the heritage and the lands that originally were home to First Nations, the lands on which CAMH is on. And I don't know if you got a chance, but it's like a Native American month, and there were very interesting things on the radio as well, like stories and music, so like definitely it's very interesting to see all the things that can expand our horizon. So we're definitely honoring and respecting the tradition. All right, so in terms of synopsis, we'll have 2 sessions. The first one would be more about microcircuit activity in mental health. The second one will be more about EEG signals. Then afternoon we'll have tutorial sessions that roughly are equivalent to that content. I will present lectures, and Frank from my lab will lead the tutorial. Frank will be available during my session and also during the break to assist anyone if need be, but I will encourage you to focus on listening to the talk. I think between the talk and the tutorial there will be time to set up things. There is the binder, which is a really simple way to get the tutorial code to run. It really doesn't require much, so that should be relatively smooth to set up. All right, so with that, let's dive into the material. So, as a brief overview, we model cortical microcircuits. So those are networks of neurons, not huge ones, but thousands of neurons that can do quite a lot. Let me just change the cursor here. Right. So that would usually correspond to a brain region, either a layer within the region or the multiple layers within the region. And usually there is a function attributed to that region. We study those microcircuits, those are detailed microcircuits in depression, schizophrenia, and aging. And there are 3 ways that we use those models. The first way is to understand how brain function happens in health and disease, mainly processing information or stimuli. Here you see an example where you can essentially, most of the things that the neurons are doing are extracting features. It can be about simple features like parts of an object that you see. It can be more complex features, like some meaning in a sentence, but the idea is roughly similar. Another way that we use the models is to try to see, are there signatures of the changes in circuit activity that we can see in clinically relevant signals like EEG. because we have detailed microcircuit models, we can also simulate the signals that arise from them. So we'll talk about that. And the other thing that is really cool, but I'm not going to talk about too much today, is we can also simulate drugs and treatment protocols and see how, characterize them very systematically in silico. And that's something that if one has to do experimentally, it's either impossible sometimes because in humans our ability to do things is limited usually, but also it takes many years. So computational models and detailed ones offer a really good complement for that purpose. So the first session, we'll talk about simulating brain microcircuit activity in mental health. So the previous day you heard about genes, a lot about genes, and that's sort of where we often start, right? I think one of the nice things that computational genomics can give us is candidate mechanisms that are altered in diseases. For example, let's say there is SST expression in interneurons that it is reduced in MDD or depression. Then we get to the point that we ask, okay, what is the meaning of that? How does that affect brain function, right? And the nice thing, and I think maybe that's the main thing you want to take home maybe from that level of research, is that neuronal networks in those microcircuits code something. And we're at the level where we can really ask direct questions about the neuronal coding and the impact of changes in disease on neuronal coding and how it can translate to function. So that on one hand, on the other hand, obviously we're limited to a small scale, but remember that that's the pro of looking at the detailed neuronal networks and their spiking, as opposed to more simplified representations of brain areas to look more at their activity. So I think that's one thing you want to keep in mind, and that's you're gonna see throughout this session how we take advantage of that. So let's start understanding what are those microcircuits. This is a term, right? So it refers to a part of the cerebral cortex, usually on the order of less than a millimeter cube. And that part of the cortex has several layers. Like the cortex is divided to 6 layers. So sometimes we model the whole column, as we call it, of layers. Sometimes we model just a specific layer, for example, layer 2-3. For example, in humans, we have much more data about layer 2-3. So just if we want to stay data-driven, we stick to that layer. And also because it's a functional computing unit. So there are thousands of neurons in each layer, and they on their own can do a lot. Interestingly, like the number of neurons on such a columnar dimension is about 30,000 neurons. And that's true both for the rodent and the human. So it's not like humans, even though the brain is bigger, you have much more neurons. It's actually the density is lower. So the number of neurons is actually the same, even though they're larger, each one of them. And those microcircuits have very specific types of neurons and also very specific connectivity statistics and properties between them. So that's really what defines those microcircuits. All right, so a few words about what those microcircuits are doing. The simplest thing you can think of is really detecting features about stimuli. You know, the sensory microcircuits have been maybe the most studied in neuroscience. And for example, harkening back to the Hubel and Wiesel experiments in cats, showing them bars of light in different orientations, and they got a Nobel Prize for that discovery. They saw, first of all, that neurons tend to be selective to certain orientations. So this neuron, for example, will not really respond to that orientation, but will respond a lot to that orientation. And that gives rise to what we call a tuning curve. And here it's for degrees of orientations, but there are actually tuning curves for even more complex features and stimuli. But that's an important way for us to probe what the circuit is doing, what is processing. So it happens on the single cell level, but also on the neuronal population level, and we'll see how that works. But that's one thing that those circuits are doing. Also, if we look especially on the population, usually you can extract more complex features by looking at the population coding. So we can simulate a baseline activity when there is no signal, and then a response across the population when there is a signal, and a readout of that response, the spikes. Here you see every dot here is a spike, every row is a different cell, and X is time. So you can read out the spikes, and essentially they represent a feature. So that's a similar idea to the tuning curve, but a bit more complex. And also importantly, you can really see or characterize the circuit output or function as a signal, no signal detection. And we'll see how that factors into some of the questions we have in depression. So some of you may have heard of neural networks as artificial ones. It's important to understand the computational power of those cortical microcircuits has two things going for it. One is the connectivity, which is specific. There are different parts of the cell that are connected by different types of neurons. On top of just the general power of connectivity, because essentially, like in artificial networks, the way that you wire your network can really help you do a certain task. So the network learns to, let's say, identify images by tuning in those weights, those synaptic weights. So that's one very important property, obviously, of biological microcircuits as well. On top of it, the neurons are very complex in terms of electrical properties. They're three-dimensional, they have different parts that have different properties, and that gives them a lot of computational power as well. So that's important to remember, and that's one of the main reasons why we bother and go to the trouble of modeling them in detail and simulating them like that. Right, so in terms of neurons, we have 4 key types. There are more and there are subtypes, but for the sake of this lecture and also in our modeling, we focus on the 4 key ones. You have the excitatory pyramidal neurons. You have the 3 inhibitory neurons. The PV inhibit the cell around the soma, the basal dendrites. The SST interneurons send axons up all the way to the distal dendrites of the neuron. And the VIP interneurons inhibit those 2 interneurons to essentially disinhibit pyramidal neurons. And each one of those cells has an important role both in baseline and response, and we'll talk about some of it. We get those are human neuron reconstructions, by the way. And as you heard yesterday from Srijoy, it's nice that there is increasing data on neurons and synapse and circuits from human tissue because they can be extracted during surgeries. The tissue on top of the focus of the epilepsy or the tumor for which the surgery was done is much more healthy. And has been characterized to be healthy for all intents and purposes, and that can be used to do experiments and record from the cells and everything. So that's really where our data starts. And we have collaborators that Srijoy mentioned as well in Toronto Western, but also there are public databases where you can just go online, like the Allen Brain database. You can search for cells. You can ask for human, for mouse cells. You can look at the donor profile if it's relevant to your questions as well. There are a lot of stuff there, and it's all free and very nice interface. So that's where we get a lot of our data, and that's a very commendable open science venture, which we really appreciate. That's an example of results. Let's say that you looked for human pyramidal neurons, right? Then you get them. You can see the recordings. You can also see morphologies and where they were. So we would usually search and find data that is good enough, because there is always 1 or 2 or 3 cells that are not as good, but they're still in the database. So we do spend time looking and curating things, and that's also where Shrijo and his group are a big help for us. For example, they curated a lot of the data from our collaborators, and then we could trust it more and use it more easily. And then we set up to model the cells. And one of the things the key things that we want to usually model in neurons is their firing. So traditionally, the firing in the cell is characterized by giving it a step pulse, and then different cell types will have different characteristic patterns. So for example, pyramidal neurons will tend to burst in the beginning and then fire a bit more regularly. SST interneurons tend to adapt their firing rate as the pulse progresses. So we want to capture all of that usually. And the other important thing is the frequency input curve. It's like the input-output gain of the neuron. So how much would it fire for a certain current amplitude? So for example, you see that pyramidals are less steep in their response compared to SST interneurons, and PV interneurons, which are also called the fast spikers, are even steeper. So we want to capture all of that. That's the data objectives that we start with. And the approach that we usually take, and you'll see, I mentioned why, but you'll see even more later, is we use detailed models that have the electrical cables reconstructed, essentially, all those dendrites of the neuron. And so it starts by, there is a reconstruction that the experimenter is doing under the microscope and reconstructs it in the computer. And then our software called Neuron converts it to cylinders, segments, essentially. And each cylinder is broken into compartments as well. And compartment is really the smallest piece of electrical unit in the cell, which is represented and computed in terms of voltage. So that's really a series of axial resistors, which really represent the conductivity along the cable, and also capacitors and resistors that represent the leak currents across the membrane and the membrane itself, which is really a capacitor. So that's like a electrical circuit representation of this neuron called compartmental model. And on top of this, we distribute ion channels. So those ion channels are conductances across the membrane. They have a reversal potential depending on the ion. And they're represented by differential equations that really started by the very seminal work of Hodgkin and Huxley. So they found ways to characterize the sodium channels and the potassium channels in terms of gates, opening and closing, and those kind of equations. They got a Nobel Prize for that. So we use the literature mainly to look at, get all the channels, the different channels, and their representations, and we can model them and add them to our cell models. So those equations govern the inactivation, activation, and the time constants of those channels. It's fairly simple but detailed and very powerful. So once we have those, we can simulate a cell. And here you can see there is a video simulation of a spike starting at the soma and then propagating to the dendrites. We can see it once it starts again, essentially. But what we do is we essentially distribute the ion channel across the cell, especially around where the action potential starts, but also all the properties along the dendrites. And then we can simulate really the electrical events that are happening in the cell. And you can see now there was a spike, it quickly backpropagated to the dendrites, and it's a lot of equations that are solved, and Neuron does a great job in solving it. It's a free software, by the way, and it's very nice. So what we don't know usually to a good degree is what are the densities of those channels in the different parts of the cell, and for that we search the solutions using algorithms. So essentially, we know roughly what type of channels. We sometimes know roughly what's the range of the densities, but we don't know exactly what are the densities that can reproduce the properties you see in a certain type of neuron. So in that kind of optimization, I'll show you in a sort of like a nice sweep through sort of a taste of how we do that kind of optimization. We start with data. We have features of the spikes like height, width, the rate of the spikes, the adaptation, and we get mean and standard deviation statistics. And then essentially we run the genetic algorithm with the idea of also multi-objective optimization. And a quick word about it is that instead of weighing all the features into one cost function, we treat them independently and use the idea of multi-objective optimization to rank based on dominance. And the idea of dominance is as follows. If you have two features, let's say spike rate and adaptation, also called accommodation, then you look at all your models. Every circle here is a model. And then what you want is essentially really the models that do best in all, right? But part of the way that you get to that point is you also rank models based on whether they're doing better than other models. So for example, this model here has a low rate error, right? Because it's right here. It does better than all those models. Essentially, all those models have the same accommodation error, but a worse rate error. So in a way, this model dominates all those models. So if you had to select a model, this one is better. The same way, this one dominates in terms of accommodation because it has a very low error. Those two don't dominate each other, so you want to keep both of them. But this one does better than both of these. Or actually more accurately, you would say this one is not better than that in one feature, and this one is not better than that in another feature. So you would want to keep that as well. And there is that theory that this front, where all those models are really not dominated by any other model, is your Pareto front. That's where the kind of models you keep. And it's a very powerful concept. Obviously, I just gave you a teaser about it, but you're welcome to look more into it. When you have a lot of things you want to fit at the same time, this is a good way to not have to weigh them all against each other. You actually just try to get the best overall performance independently in all of the objectives. So that has served us really well in fitting those very numerous features of the electrophysiology of the neurons. So also a quick explanation a bit about the genetic algorithm, because it's also, I think, could be useful for many of you for optimizing or fitting other kinds of data, I'll give you in a nutshell the different stages of it. So you start with the population of models. As we said, our search is for the densities of the different channels. So we have all those parameters like the sodium transient channel, the calcium channel, a potassium channel of one type, and then another type. And you have a lot of models, let's say n models. And each value is the channel density here. And you can look at it almost like chromosomes and genes. And the idea of genetic algorithms that are also called evolutionary algorithms is that they use mutations, crossover, and selection, like in natural evolution, to gradually converge and really get to the best models. Really the same way that natural selection improves the survival and the fit of individuals. So the second stage, you can allow some mutations. So you pick some values and you mutate them with a certain probability. So for example, model 2 got a bit of a mutation here, switch from 0.2 to 0.18, and also in this parameter. And what we usually do, we start with large mutation amplitude, and we gradually, as the iterations go, we decrease it, and that some of you may know, it's called simulated annealing idea. On top of it, we can also allow some crossovers. So essentially model 1 and 2, like chromosomes, can switch segments. When they, this is essentially, they can sort of inform one another or switch. And then you see this whole segment from model 2 switched to model 1 and vice versa. After we do this one generation mutations and crossover, we evaluate the fitness of the models against the data. Again, to emphasize, this is a very data-driven approach. It's automated as well. And then we get errors in terms of standard deviation from the mean. Now, an important word here is that a population of pyramidal neurons is not the same. They have some variance in terms of what's their spike rate and all of that. So it's not like the model that has 0 deviation from the mean is the best, right? But there is a certain range that we want to allow. We don't want some things beyond the physiological range. So if, for example, once we rank the models, the ones that have a large deviation, we tend to then throw. This mutation, by the way, and crossover creates a new population on top of the old one, so we end up with 2n models, and then we select n of them to start the next generation. And like I said, for example, we would throw that model because the deviation of it is so big that it's becoming non-physiological, really. And then we iterate until we have convergence, all right? So that's in a nutshell the genetic algorithm. People have actually also used it to fit networks, to play Atari. I mean, some of you may know of the deep learning networks that play Atari, but people have also, from Uber, published a nice network that was trained using genetic algorithms. So those are pretty neat. They're very useful when your solution space is complex and you can't really solve it in terms of equations and learning rules, right? Because what is really the learning rule to search through those densities, right? Here it's fairly hard to do it with deep learning, really, when you think about that. So that's very useful. And there is a lot of models in neuroscience fit using that kind of approach. Obviously, one caveat is that it's sort of more of a search. It's not like optimality is guaranteed here, but usually the convergence is good. So it's not a big issue. Simulating all those neurons in detail, and like thousands of them, usually takes a lot of resources. So we usually use parallel computing for that. For example, on SciNet, there is NSG as well, and in the past I've also used BlueGene, but SciNet is really the public resource that is relevant for us here. And there is a nice package from the Bluebrain project called BluePyOp that is open, and really this whole algorithm is provided in that package. So I really recommend looking at it. It's been really useful for us. All right, so after we fit the models, we end up with models that reproduce key properties of the neurons. And here you see from our lab, we fitted human neurons. So this is a human L23 pyramidal, L23 meaning layer 2-3. So it reproduces the typical pattern and also the response to hyperpolarizing currents that are a good way to characterize certain currents that stem from the dendrites called the SAG current. So this is what you see here. The SAG current is that a small blip on top of the hyperpolarizing current that really stems from a lot of current coming from the dendrites, which affects input integration. So that's an important thing you do want to capture in your models usually, because then you have a good model of how inputs are summated and integrated along the dendrites before they reach the soma and the axon. Those models reproduce that input-output curve that I mentioned. So what we usually have, as I said, is a population data. So that is demarcated here by the shaded area from usually 20 to 30 neurons. And so these are the standard deviations, and the experimental mean is in light gray here, and you can see the model sits nicely on top of it. But as I said, any kind of a curve within that range is pretty physiological. So we do want to stay within that range. And that's an example of an SST interneuron. Now, interneurons, there is much less data about them, so usually we have just a few cells at the moment, but I am expecting that in future years that will change. But that kind of approach can also be used when you don't need a population data necessarily for that, so we can match a single neuron. So we have essentially now models that reproduce human neuronal firing and integration properties. And a word that is worth mentioning that is nice, the genetic algorithm usually gives you a population of models. And this is important because studies have shown that the same way that not all neurons of a certain type are exactly the same, they also don't necessarily have the same channel densities. And if you want to think about genes, you can think about gene expression as well, right? So different combinations can be seen in different pyramidal neurons, even though they're roughly similar as a type. So a nice thing that we get from the genetic kind of optimization is we get a bunch of neurons. This is an example of like more than 100 neurons that were fit for a layer 5 rat firing. And we can use those solutions, the models that are physiological, to delineate the ranges of densities that can reproduce the physiological properties. So that's very nice, both to understand changes, because you can compare different cell types and the ranges, and also to relate it maybe to gene expression as well. And for example, if we compare, in this paper, I compared not two different cell types, but two different kind of models, ones that didn't care about dendritic properties in black, and ones that did try to reproduce the dendritic properties in red. And you can see that the necessary densities are much more tighter in this case. And for example, the density range that we estimated using this really simple just analysis of the population of models that are spit out by the algorithm, the range is very in line with what is estimated experimentally for dendritic sodium channels, for example. So that's a very powerful tool automatically to generate a lot of models and also then have a population of models that can correspond to the physiological population, and you can ask questions about different conditions, models. You can have like 1,000 models for depression, or let's say, what's the best example we can think of? In a certain condition where you think there is a change in those channels, it could be aging, let's say, right? Then you can compare those two population of models and come up with some predictions. All right, I'm gonna, I think, pause here because I feel like I'm talking to the ether a bit. Frank, how's everything so far?
Everything's going well.
Good. Are there any questions, or should I?
No, but I encourage everyone, please post any questions. Etay is very knowledgeable. Be happy to answer any of them.
Yeah, so feel free. I'm sort of, my plan is not to rush too much, but obviously we started a little bit late, but I'm happy to dwell on a few things if need be. And also, I will, I do want to sort of go in a nice detail about certain things. So I hope the pace is okay. All right. So let's talk a bit about the next step. Now that we have models of the individual neurons, we want to connect them. So there are two types of connections. The first type is the excitatory type. And to model it, you model several things. First of all, you model the connection site number. So how many contacts, synaptic contacts, are there on the postsynaptic cell? Here you see a pyramidal to pyramidal. And sometimes the number is small, sometimes it can be quite high actually, like 20. So that's important. Another thing is that those synapses are actually stochastic, and we have it in the models, but I'm not going to get to it here. And the last thing is that you want to also represent the amplitude of the EPSPs and also the short-term dynamics of them. So each type of synapse tends to summate a bit differently. Here you see that they don't depress too much or don't facilitate too much. So that when the presynaptic cell fires a train, the postsynaptic cell will respond like that, and we want to capture that. And I'll mention that this data is available for human actually in recent years, and we've been able to use that to fit. So it's nice. There are labs that record pairs of neurons when they stimulate one neuron and then record from the other neuron. Another types of synapse, which is very important as we'll see for depression models, is the inhibition. So let's focus on this part first. Essentially, an SST interneuron synapses in quite a few locations on the pyramidal neuron. It has very strong synapses, as you can see by the amplitudes here. And we model that and capture. So the experimental mean is here. And if we simulate different trials of synaptic events, we capture that mean and also the variance. Another thing, essentially, that this inhibition is mediating a blanket of inhibition in the cortex as follows. So essentially, a pyramidal neuron firing can trigger spikes on its own in SST interneurons because the EPSPs that result from that firing on the interneuron summate very strongly. So after that interneuron fires, it inhibits a neighboring pyramidal. So what happens when pyramidal neurons fire, they create a feed-forward inhibition on other pyramidals to tone things down. So that's an important property, and nicely enough, people have characterized it in human tissue, and we were able to use that as well to model. So those are two examples of the types of synapses. There are synapses between PV interneurons. Let me just go back to this diagram. So we talked about pyramidal to pyramidal. There is SST to pyramidal, pyramidal to SST. As you can see, there are a lot of combinations here. Nicely enough, there is quite a lot of data from human tissue, which shows that the inhibitory synapses are stronger than in rodents. So that really gives us a motivation to do human-specific models, because there are some key differences, and I'll list them a little later. But I'm not showing all the possible connections, but I can mention that we also have good constraints from human tissue for PV to pyramidal and pyramidal to PV. So quite a lot of their circuitry has data in humans. After we have that, we want to distribute the cells in a volume with the right proportions. So each cell type has different proportion, and also depending on where you are in the cortex. So in somatosensory microcircuits, you have 20% interneurons, 80% pyramidals. Pyramidals are the most predominant cell type. In the prefrontal microcircuits, you have more interneurons actually, and you see it both in humans and rodents, and that's very interesting. So also the proportions can differ based on the layer. So layer 5 might have different proportions of cells. So we want to have the right proportions. And also we want to have the right connectivity statistics. Some of it we have from humans, and some of it we have from rodents, essentially from the Allen brain and blue brain databases. A lot of, like, thousands of experiments between many combinations of cell types. They give us what is the probability that each neuron of a certain type is connected to a neuron of another type. For example, for humans, the connection between pyramidals is 15%. So it's just an important example. It's not an all-to-all connectivity. It's not like a pyramidal is connected to all the thousands of pyramidals that you have in layer 2-3. It's connected to a certain percentage of them. So that's important to capture in the models as well. All right, so a quick summary, as I promised, to some of the differences between human and rodents, and essentially the motivation, why do we want to model human? Because with rats, we know a lot. But first of all, human neurons are larger, and there have been nice studies that show that affects the input integration, because those distal parts are much, much, much further than the soma. So the inputs decay more by the time they get to the soma. So the whole input integration is a bit different in humans. Another thing is that inhibitory synapses in humans are stronger. So that also affects the balance in the circuit. Recent studies suggest that maybe the excitatory are strong as well, but it's still not yet established. So I'm not going to say more on that. But the main thing is that it's not necessarily the same as rodents. Another thing is human cortex is just bigger, not just the cells, but the whole dimensions are bigger, and that can affect the contributions of different cells and different layers to the EEG signal. So that's also something, if you want to model circuit in the cortex and also the EEG signal, that being human specific might be a bit more accurate if you want to translate your simulation predictions to human EEG. And consequently, it has, as I said, better prediction power for human disorders, because, depression, we do prefer to study it in the context of humans, both in terms of symptoms, both in terms of circuit property and activity and signals. Having said that, I also mentioned that there are some similarities. So nicely enough, I mean, a lot of the circuitry motifs are similar. So we can leverage a lot of data we have from rodents where it's missing from humans. Right. I'm gonna pause here now. Let me just look for a second. Right, the chat box with questions is empty, which is fine. I can see it here. Everyone is okay? Thumbs up. Right, I'll take the silence as a relatively positive thing, maybe. Frank, we're doing okay so far?
Yep.
All right, so I'll continue and share my screen again. All right, thank you for the thumbs up. All right, let's share the entire screen. And we are back on course. All right. So now that we have a circuit with the human neurons modeled, the human connection, the synaptic properties modeled, the statistics of the connectivity modeled, we want to start activating it. So what we usually do, we give it some background input that represents a cortical and some low thalamic drive to get it going. It's not a strong input, but it's a necessary, and it's the physiological way where those circuits start their recurrent activity. So we do that, and then the main thing, the first and very simple thing we ask from that circuit model is for it to fire at the right frequency, more or less, the baseline frequencies. And for this, we have some data from humans because people have recorded unit activity extracellularly during surgeries, especially in different parts of the human cortex. So we do have that numbers. For neuronal types, we usually don't have much. There is some data for PV because they are easily identifiable even by extracellular electrodes. But we do harness some of that data from rodents as well. There is a good equivalence between the two in terms of baseline activity. So as you can see, when we do that, we get activity in the circuit. This is all simulations here. You see about a second or so. This is a raster plot. And you see every row is a cell, every tick is a spike. It's color-coded according to cell types. You see a lot of pyramids in black, obviously. You do see events where they tend to be a bit more synchronous, followed by some interneuron synchronous activity. And we can get the models by tuning the background input and a little bit the connectivity to get them to fire at the right levels. And that's what you see here. And just to mention, a lot of this is work done by Kent Yao from our lab. Also, it's a teamwork, but we have a bioarchive on that you can refer to for further details. So now that we have those healthy microcircuits, we're getting to the mental health questions, right? So we have a pretty useful and realistic tool that we can start probing. Right, so let's remind ourselves of where we started. There seems to be changes in depression. Just a bit of background first. Obviously, MDD we refer to as depression. It's a major depressive disorder. It is a leading cause of disability in Canada and worldwide. And unfortunately, a large percentage of patients are resistant to the SSRI treatments. And various studies in the recent decade or so implicate altered inhibition as being associated with treatment-resistant depression. So there are TMS studies, there are some other kinds of studies that really show that inhibition is probably reduced, especially in those patients that are not responsive to serotonin modulation. Right, so more specifically for us, and that's going back to where we started, gene expression studies can suggest mechanisms for us to probe. So one very important study from Etienne Sibille's group at CAMH looked at postmortem tissue in healthy and depression patients and showed that if you look at those SST interneurons and you check their expression level of SST itself, there is a reduction in depression compared to control, about 40%. And it's seen across all layers, but for us, especially in layer 2-3, which we model. And this involvement of SST interneurons as a mechanism potentially underlying the reduced inhibition in depression is supported also in studies in rodent models, where they silenced those cells or activated those cells and saw stress-related changes. And also when they apply pharmacology that acts on those synapses, they could improve the symptoms. That's a very nice line of experiments. So what we know about SST interneurons from a lot of studies in rodents especially is that they tend to play a very important role in baseline firing in the circuit. Here, those are from a paper that they recorded baseline and stimulus response in different cell types in awake rodents. And as you can see, before the stimulus, the SST is firing a lot. But then once the stimulus starts, it's being silenced. And you can see it in the spike rate histogram. And here in the summary of the rate pre and post. So overall, they seem to be more active in baseline and silent during response, especially the early response. So the idea is that if there is reduced expression of SST, it's probably corresponding to reduced inhibition. And that led to the hypothesis that in depression, if their inhibition is reduced, it's going to cause much more firing in pyramidal neurons at baseline primarily. And if you want to look at it in terms of function of the circuit, there's going to be much more baseline activity and not so much of a change in response activity. And you can see it as a change in the signal to noise ratio, because essentially it makes it harder for you to say whether there was a signal or no signal just based on reading out the pyramidal activity, because it's becoming more similar in depression compared to health, where you can clearly see, okay, there is something going on here. There is nothing much going on here, right? So this is the SNR hypothesis, and one of the things that can be done using those circuits, and we've done it as one of our main projects, is to test this hypothesis. And it's important also to understand, part of the question is, can the level that is estimated by gene expression, let's say there is a 1-to-1 relationship between reduced expression and reduced inhibition of about 40%, does that lead, or would that lead to a significant change in the firing and therefore the SNR? And interesting connection also to another symptom in depression is rumination, where you can look at the increased overactivity at baseline when there is no signal really as really contending with or sort of promoting self-referential activity that could maybe play a role in explaining rumination. So the role of SST, as you can see, in depression is very interesting and promising. And as I mentioned, there are drugs that have been developed at KCNI that counter that reduction that seem to be really promising new treatment for depression. But still, as you can see, many things are not as well established and as known mechanistically. That's exactly where the circuit can play a role. So what we do in that case, we use that gene expression data to suggest a certain modulation or change in the circuit. We take the healthy circuit and we change it. And what we do is we change the synaptic inhibition, but also a thing called tonic inhibition. It's more of a steady inhibition that is also mediated by those cells. And then we try to see, okay, what are the effects now on the circuit activity and also on the function? So we start at baseline. You see here a simulation of a healthy circuit and a depression circuit. And visibly, you can see the depression circuit is much more active at baseline. So there is no input really to that cell, like in terms of external, but it's generating much more activity. And if you look at the dose response, as you decrease SST inhibition more and more, you do increase that baseline activity. Here is the healthy microcircuit, here is the depression microcircuit. There is about 50% increase in baseline activity. So the next step, right, to understand the implication on the function is also to look at the response. And we can model response using different studies, especially from rodents, because that's where more data is available for the different cell types. But keep in mind that canonical contributions of different cell types to response seem to be preserved across species. So the data that we use is the rates in baseline and response in the different cell types and also the profile of the response. You can see that pyramidals tend to respond to that brief stimulus by a brief activity, and then it tones down more later. PV and VIP interneurons respond with high rates as well, but SST, as we mentioned, are silent usually during the early response. Another thing we take into account is the activation of the different cell types in relation to when the stimulus happened, and that data we have as well. So once we put it all into the circuit, we can activate the different populations to reproduce the kind of response profiles that you see. So this is the pyramidal brief peak response after stimulus here at time 0, the dashed line. And then it tones down to some kind of recurrent activity. So you can see here we activated this small group, but then the activity propagates to the entire pyramidal population. You can see obviously the difference between response and no response here. This is the baseline, more low rate activity. And here you can see the activity in the other cell types. And as you can see, the red dots, which are the SST, are pretty much absent here because they are silent. So once we have a good model of response, and this is really simulating a neuronal network being activated by any kind of stimulus. This is, you can look at it more of a generic model of cortical processing, even though it's obviously constrained with specific data. We know a lot about the sensory response, but conceptually, it's very similar across different parts of the cortex, at least to brief stimuli. So we can compare how does the response and the baseline are in depression and health healthy microcircuit models. So the healthy ones are in gray, the depression are in purple. You can see as before that there is an increase in baseline activity in depression. But because the SST are not playing much of a role in response, they don't really, the fact that we reduce their inhibition doesn't really affect response. So what you can see is that the baseline rates in depression are approaching closer to the response rates, and that reduces the signal to noise ratio overall. So that's a nice measure, but we were interested to interpret it a little bit more rigorously in terms of functional meaning. So what we did is the following. We looked at the pyramidal neuron rates, because you can think about of the circuit as a circuit that does something that is then being read out by some other circuit down the hierarchy of the processing. So if you want to ask what is the meaning of a certain change, let's say depression, you want to ask yourself, what is the change in terms of the functional readout? And for this, the pyramidal neurons are the most relevant. So we look at the baseline firing here in that dotted curve. That's the distribution of the pyramidals firing at baseline in health. And this is in health, their response rates. As you can see, obviously, there is some overlap, but also some good segregation. And then we can quantify the false negatives and positives. So if you had to say whether there was a stimulus or no stimulus based on the pyramidal activity, then you would, according to theory, would put the decision line in the middle here, where the two curves intersect. And that gives you an ability to quantify failed detection and false detection. So essentially, here you would say there wasn't a signal, but in actuality there was a signal, right? It's just that your decision boundary optimally is put here, so you're missing some of the signals here. And here you would say that there was a signal, but there wasn't a signal, right? So that gives you some metrics that are actually even relevant because they can be measured, and they have been measured in depression behaviorally. And when you compare it to the depression microcircuits, you see that those failed and false detection rates are much higher. You can see it by that shaded plot. And the reason, as we said, is because the distribution of the baseline firing rate is getting closer and closer to the distribution of the response firing rates. And here it's quantified just by the rates themselves, what is the percentage of failed and false detection. You can see there is a 2 to 3-fold increase in between health and depression predicted. And nicely enough, that matches experimental measurements that have been done in depression. They did a continuous performance test where they show people letters and they need to respond as fast as possible. And they did see omission and commission errors, which is exactly the failed and false detection rates. So it's nice. I think that's sort of going back to where I started that session. This is really the things we want to get from the circuit, not just the activity, but also like what is the functional meaning of it. And by simulating neuronal networks that are spiking and are coding for something, we can relate the output of the simulated microcircuit to behavioral metrics that you can measure. So that was a good example of how that can work. So to summarize that part, one of the key things to take home of the applications for computational detailed models of microcircuits, the first thing is to establish target mechanisms for new treatments. So for example, if you have a mechanism that your gene studies or other theories or other studies suggest, is underlying a certain disorder, and you're fairly limited in actually showing the cause and effect mechanistically in humans because experimentally we're much more limited. The computational models offer a very powerful tool to complement the experiments and guide future experiments with concrete prediction and more mechanistic understanding. So I think that's one of the key things where I see the role of those circuits, right? I mean, it's really helping establish target mechanisms and really understanding what's going on there. Another thing, and that harkens a bit to the clinician talk that we heard that was very fascinating from the first day, all of that stuff can really complement other things that you know about patients to help you better as a constellation stratify them, right? Because, for example, if we show that people that have more false detections or failed detections are more of the SST type as opposed to maybe the serotonin-mediated type, then that can really help understanding the underlying mechanisms, the type of deficits, and that can help also understand other deficits that are related. So I think those two are probably one of the key things in terms of applications where those in silico simulations of the brain circuits is really playing and can play a nice role. Yeah, so that is the end of this session. I will stop sharing now. There'll be obviously time to ask questions later as well. I see that we do have a few questions. The second session, it's nice that we're actually roughly on time. The second session I can also use to answer some questions, but we can start. What level of differential equations knowledge is adequate for this type of research? Kevin is asking. That's a good question. I'm going to, in the second talk, wrap up with some kind of big concepts, and I'll mention some simpler models, but I think it's worth also mentioning now. You can model those circuits in a different degree of detail. You can use fewer compartments to represent a neuron. You can use one compartment to represent a neuron. I think the key thing here is the spiking. I think what maybe differentiates this level of looking at things than maybe a more of a population dynamic kind of mesoscale is the fact that those neurons are actually spiking. You can ask, what are they coding? So I think the level is actually fairly flexible. I would say that the advantage here of that level is that you're not, when you want to try to simplify things, it's a big question of what are you losing, right? What are you missing? And here, we don't need to get into it. We have the full detail. The neurons are as electrical entities as they need to be. The inputs are integrated in different parts of the cell as they are actually. So that's one advantage. Bottom line, like I said, there is flexibility, but also definitely some advantages to having that detail. But it really depends on the question. You can do both. I hope that answers the question. So I've started answering and I'm done answering. Somebody, Reza, put a question in the chat. I'll get to it after. Question by Yudajit. Would the low SST expression in MDD reflect in frequency power characteristics in EEG, higher power of background frequency? That's a great question. That will be in the next talk and the next session, where we capitalize on the realism of the models to simulate EEG and try to see, do we see biomarkers of that? So I'm not going to say more, but that's obviously an important question and a good one. Question from Reza. Speaking about circuits and modeling, either Amos, volts, resistant potentials, or a combination of them may play a role. I appreciate your insight. If this possible role is equal or should be adjusted and weighted based on the experiment. Okay, let me just see. For example, where cells respond to certain currents induced by physiological or pathological process, how do you know if your model is fit for further experimental studies towards possible clinical trial? Okay. Some of the points I'm getting from this question is essentially how do we, like the constraints of the models and maybe generalization, right? So we do constrain the model to reproduce the right response for the right current input, because we use those experiments where you inject the current with an input and see the response. And so a lot of it is actually very nicely constrained in terms of electrical input and output. In terms of generalization, that's another nice thing. They do tend to generalize nicely to experiments for which they were not constrained with. And we'll see it in the second talk by the fact that the circuit actually reproduces a lot of features of EEG that you see in humans in vivo that we got without specifically asking for them. They just emerged from well constraining the different properties of the circuit. But that's a good question. You do want your model to generalize well, and yeah. Next question from Alexander. Possibly a discussion point that may be of interest here. How does one go about deciding whether or not a model is detailed enough or too detailed for the research question being tested, e.g. how to choose a model that balances the computational load versus model complexity versus being able to clearly decipher your results, etc. So yeah, that's a good comment, similar to what I said. I think nicely enough we are flexible, and it's good to pinpoint what is the important part of that level of looking at the brain in relation to mental health. And as I mentioned, this is, I would say, the fact that you have a spiking neuronal network is one of the more important things. But keep in mind also that you do want to have the right detail to be relevant to your mechanisms. So if you have a gene expression change in SST, or if you have something in Alzheimer, you do want to be able to model it. And you can model it sometimes abstractly and simply, but sometimes having that detail really gives you a very easy way to, because it's already there. We have SST, it's connected to pyramids and the rest of the cells in the right way you see in the brain. So any changes you do to it has a lot of prediction power in terms of what can go wrong. All right, question from Povilas. What are some possible mechanisms that can lead to the reduction of SST? That's a good question. I think that also relates to, what stages of depression it is, right? Like what is, how did it come about? When does it come about? So maybe a few words about it. The first thing is that it's still a work in progress in terms of understanding that, but I do know that it is related to early stages, especially after there has been a chronic stress. So a lot of the models animal models that the TN uses to understand the circuitry and the effects are stress models in rodents. And they show that a lot of those changes need some time. That's not an immediate thing. So you can, I think we do see it as chronic stress, but more as early. But SST changes also in aging and in some other conditions. I think it's a very good question, which unfortunately remains to be answered further. Yeah, that's as best as I can say. All right, I think I'm done with the box here. Yes, and one more question. How would you conclude that your in silico model is best fit for your further experiment? Yeah, okay, I think I answered that. All right, so we're actually right nicely on time. We could wait a few more minutes in case someone has a question. But we will be soon taking a break for 15 minutes until 1:45. I hope that you learned a bit. It's funny, this year we have a bit more time, so we can get into more detail, but obviously a lot of those topics are a little deeper, and I suggest and recommend, people can go over the slides again. I put some references, and you can look further. Okay. Right, so Frank can hear me, but I think you can. Hopefully. Good. Yeah, it is a bit challenging. It would be nice maybe next year to do this in person, where we can actually see the crowd, and it's a very interesting experience to talk to the ether and hoping that everything is actually being transmitted. Good. So I think I will take this point as a break. Thank you, John. And yeah, I will see you in the next session. I will end the broadcast now. Enjoy your break and see you in 15 minutes.